So in the short amount of time that I have, I just want to make 2 points, I suppose. So what we're doing broadly in the lab is integrating genetics with herbicide studies to try to improve water milfoil management outcomes. And so this is sort of broadly across the landscape. And one really important point is that there's a lot of genetic variation for herbicide response. So not all Eurasian water milfoil is the same. I'll just show a couple of really quick graphs to illustrate that. So here's a dose response curve of several different strains of Eurasian water milfoil. We've got fluridone concentration on the x-axis. Fluridone's fairly commonly used in some areas to control Eurasian water milfoil. And then on the y-axis is growth relative to a control. And so we have some strains that are really highly resistant to fluridone, like this strain here. We have strains that are really susceptible to fluridone. Fluridone would typically be used for Eurasian water milfoil in somewhere around the 6 parts per billion range. And then we see a lot of variation in between that. And we don't really know how we know how this variation translates to the field. We know how this translates to the field. We don't really have a great sense on how some of these other things translate into the field. Just one other example I'll show. So this is also a dose response curve. This is for 5 different strains against 2,4-D, which is another really commonly used herbicide to control Eurasian water milfoil. And so you can see we've got some strains that are more resistant or less susceptible to some typical concentrations of 2,4-D used. So in the 500 parts per billion to 1 part per million range, whereas others are quite sensitive to that. So although many factors influence management efficacy, right? There's lots of hydrological things, there's chemical things, there's biological things, there's technical things. Genetic variation within and among lakes is clearly an important factor. And so ideally, we would know something about the strains or the genes that we're treating so we can make informed herbicide decisions. So in our program, we have a short-term approach where we're building a catalog of water milfoil strain, herbicide response, and geographic distribution information that's publicly available. I'll show you that over the next couple of slides. And then in the long term, we're identifying the specific genes for resistance so we can develop genetic tests for those genes. And I'll spend a few minutes, hopefully, on that as well. So we built this little publicly available app. It's called Milfoil Mapper. This was done by Ashley Wolf, who's in my lab, and then Alex Badges, who's at the Minnesota Aquatic Invasive Species Research Center. So this is the link here. If you want to copy, I can also get it to you. You can also just Google Milfoil Mapper, and you should be able to find it pretty easily. It'll take you to an app that looks like this in the home screen, and you can query this database basically by geography and strain. So you can pick a lake, you can pick a county, you can pick a state, or if you know a strain that you're interested in, you could query by that strain as well. So these are all the locations, or at least in the sort of more eastern half of the U.S., where we have water milfoil strain information for currently. And so here's an example where we picked a strain to query, and this strain is fairly common in the state of Michigan, and it's an interesting one because it's resistant to 6 parts per billion fluridone, which is like the common fluridone treatment in Michigan. And so it'll show you the distribution of that strain. It'll print a list, and then if you click on a lake, it'll give you a list of all the strains that we've seen in that lake. And if we know anything about their herbicide response, it'll tell you something about that too. So it's a little difficult to see. I apologize, but there's 2 strains here. One that we don't have any herbicide response information on, and the second one, which is the one that we queried here, has been deemed as susceptible to 2,4-D, but resistant to a typical Michigan operational fluridone treatment. So I'll forward through this in the interest of time. But so at any rate, for those strains that we have characterized, they can directly inform management decisions. So here's an example of a strain that's really widespread. This happens to be a pure Eurasian water milfoil strain, by far the most common strain of Eurasian water milfoil that we come across. And we would have no concern about using fluridone where this strain is dominant because we characterize it in the field and in the lab as susceptible to fluridone, in contrast to the strain that I just showed you before. And so in states like Michigan, we're doing a lot of screening of lakes, genetic screening of lakes before treatment so that, we can see if we have any information about whether or not fluridone would be a good tool or not for any particular lake in Michigan. And we're sort of expanding that more broadly geographically. I'll skip over this. And I think, you guys are probably familiar. I know Morella's done some work on the lake, so I won't get into this very much. But if you are interested to talk more about sort of the strain tracking approach, this QR code will take you to Milfoil Mapper, which has contact information in it. Ashley's the best person to contact in my lab to really consult on that and organize those logistics. So if you're interested, you can reach out to her. Okay, in the long term, though, what we really want to do is identify genes that are associated with or responsible for herbicide response. And so, using the kind of strain DNA fingerprinting that is typical that we do, that's the kind of information that goes into Milfoil Mapper. Those are good at distinguishing different strains of water milfoil, but those molecular markers don't provide any direct information about herbicide response because those markers are not in genes that are associated with herbicide response. And so the approach for Milfoil Mapper is distinguish these different strains using this simple DNA fingerprinting approach, and then characterize those strains for their herbicide response in the lab and then hopefully in the field. And so doing that, we can sort of distinguish strains like these 4 down here that are susceptible to fluoridone, and I'm just focusing on fluoridone because what we've done the most work on, versus these strains up here that are resistant to fluoridone. And so it's kind of a 2-step process, and it's effective and it's good in the short term, but it's not really where we ultimately want to be. And that's because water milfoil can reproduce sexually, and so new strains are produced all the time. So we always are going to come across new strains in the field that we haven't characterized for herbicide response yet. And so resistant strains, like these ones up here, if they engage in sexual reproduction in any lake, then they can pass their resistance genes on to new strains, and we wouldn't identify those strains based on just simple DNA fingerprinting as resistant unless we brought them into the lab and tested them. And so if we identify the specific genes for resistance, then we can develop molecular tests that can identify resistance or susceptibility in new strains that we haven't seen before. So I'll quickly subject you to the approach to doing this. So I'll broadly call this genetic mapping, and in this case, we'll be looking at fluoridone resistance. I crossed a fluoridone resistant strain to a fluoridone susceptible strain, and then what we look for is segregation in those progeny. All segregation means is a bunch of related individuals that vary for a trait. So you might imagine if you had 100 brothers and sisters, that some of you would have blue eyes, some of you would have brown eyes, and your group of progeny or your family would be considered to segregate for eye color. So that's what we look for when we do these crosses. And here's an example where we see it. So the pictures are easier. This is the susceptible parent treated at 6 parts per billion fluoridone. This is the resistant parent treated at 6 parts per billion fluoridone. These are some of the progeny from there. So some of the progeny look more like the resistant parent. Some of the progeny look more like the susceptible parent. These are just all the data here. So our resistant parent treated here and untreated, so you can see highly resistant to fluoridone. Our susceptible parent treated, and then this is the distribution of all the offspring from that cross. What we're able to do then is do whole genome sequencing of the top most resistant individuals, and I think we chose around 20 to 25, and then whole genome sequencing of the most susceptible individuals, 20 to 25 of them. This work was done initially in collaboration with some folks at the Hudson Alpha Institute. This is Alex Harkis, who really helped us assembling the Eurasian water milfoil genome, and then Josh Clevenger, who really helped us get this project started, and now we're able to kind of do this stuff on our own, which is nice. These are this is sort of all the data that come from that, and I just put it up here because I think it's freaking cool, and but it might not really mean anything, but I guess just maybe a wow factor of how much data there are. So each one of these panels is a different chromosome. There's 21 chromosomes for Eurasian water milfoil, and each one of these panels has anywhere from several tens of thousands to several hundreds of thousands of points of data points on it. And the basic premise of what we're looking for is that on average, these two groups of resistant progeny and susceptible progeny should not be very different from one another genetically because they're highly related full siblings. However, we would expect them to be different only in those places in the genome that are associated with fluoridone response, because we non-randomly put them into groups based on their fluoridone response, a group that was resistant to fluoridone and a group that was susceptible to fluoridone. So I'll zoom in on a chromosome where we have some strong evidence for one or more genes associated with herbicide response. So what we have on the x-axis is the position along the chromosome. So there's about 40 million base pairs on this particular chromosome. The y-axis, just think of it as the genetic difference between the resistant group and the susceptible group. So on average, that's going to be around 0. That's the dashed blue line here. The green and yellow lines are just the confidence intervals, 95 and 99%. So what we're looking for are points that go up above those lines. That would mean that they're unusually different from 0. The blue dots are the individual data points along the chromosome, and then the red line is just sort of the rolling average of that, so we can kind of look at the trends, because we expect this autocorrelation between data points as we move along the chromosome, because they get inherited together. So here's a spot on the chromosome where we have really strong evidence of, again, something associated with fluoridone resistance, so one or more genes in this area. Real quick, we did it with a second population, and we found, lo and behold, that same spot popping out as highly significant. And so this spot on what we call chromosome 7 really seems to be a spot that's really important for fluoridone response. And what that means is we can begin to explore that area of the genome in more detail, and ultimately develop molecular markers that are located in those regions to predict the response to fluoridone for strains that we haven't characterized yet. And so that's the basic approach to what we do. We've done that now for a couple populations for fluoridone, and we've got some stuff in the works for 2,4-D as well. And we'd like to integrate all that stuff into the Milfoil mapper application, so that which is freely available, and it's something that we hope that over time managers adopt to kind of look their strains up, or look for whether or not they have particular genes or not that we expect to be associated with herbicide response. I will, in the interest of time, because I know we're running short, I'll kind of cruise through this real quick, just to give some shouts out to some folks. Greg Troik and Del Haney have been really important in a lot of the bioinformatics work. I give shouts out to Ashley and the Hudson Alpha folks. Ray Newman at the University of Minnesota is somebody we work with a lot. And then there's been a variety of funding sources that's been going on for 10 plus years, both federal, state, and private sources as well.